So we are just a minute, I think, short of 3 o'clock. I know everybody's got summer things to do, so start, and those who join late will find us. So welcome. I am Melissa Hansen, the Research Program Director for the Washington Wine Commission. And today our WAVEx webinar focus is Dr. Thomas Henick-Kling's research project that completed last year, in which he was working with high pH wines and developing some practical treatments that winemakers can use. Just a brief background, the WAVE stands for Washington Advancements in Viticulture and Enology. And these are both webinars. Those are our WAVEx. We do WAVEx for the extension and short course. And the WAVE is a research seminar, an annual event that's gone virtual for the last year, but next year we'll be back in person. And it's a partnership between the Washington wine industry and the Wine Commission and WSU. And so this, I think, is our 6th webinar for this year. And I just have a couple of housekeeping. Remember to keep your questions in the Q&A, not the chat. We are recording this, so a link will be emailed to all of those who registered, and will also be posted in the Wine Commission's new website, our research library, where we have all the research reports. And in fact, you can find his research report in there as well. And just a pitch, we have one last WAVEx webinar next month on Brettanomyces. Dr. Charlie Edwards will lead that one. And there is the registration link. You can also, that will bring you to the Wine Commission's website. And just my last slide here before I turn this over and start my sharing. So I will stop share, Dr. Thomas, if you can pull yours up, and I'll just tell everybody. So Thomas has been at WSU's Viticulture and Enology program since its inception as director in 2009. He grew up in Germany, so he has a nice appreciation for white wines. And then he got his master's in food science and microbiology at Oregon State University, earned his PhD in Australia at the University of Adelaide, spent 20 years at Cornell University with their viticulture. And so he's really got a very broad global perspective on wine. He's worked with wine industries on both coasts of our state, and Germany, and in Australia. So we're very lucky to have him. And we're lucky that he also does field research as well as doing all his administration work. So Thomas, take it away.
Thank you, and hello, everyone. I'll give probably a brief summary of 4 years of, or a bit more than 4 years of work that we've done here at Washington State University on the impact of high pH on wine quality. I probably should start by sharing my slides. There we go. Now it should be full screen. Is that correct?
Yes.
Okay. Realizing that, this is something we face every year, obviously here in warmer climates, not just here, but in other warm climate areas, and actually more frequently around the world too. pHs in musts and wines are creeping up for a number of reasons. I want to first acknowledge at least some of the collaborators that we've had on this project. And I apologize to those who I forgot to list here, but principal collaborators were research associate Dr. Hailan Piao, colleague Dr. Tom Collins, and Jim Harvardson on the chemistry side. We had 2 outstanding graduate students, Tyler Williams and Lindsay Kronowski, working on this. And a number of undergraduate students, and I think this is where I probably missed some of the names, and I apologize. Research associates in chemistry, and our winemaking staff over these 2 or 3 years. I also want to acknowledge the support from Sagemo Vineyard and Clipson Vineyard, who worked with us to give us the fruit that we're seeking for the experiment, which amounted to quite a bit of fruit with all the fermentations that we did. And then the ongoing support, feedback from the Wine Research Advisory Committee. The whole project was funded with a batch of various pots of money. First, of course, the Washington Wine Commission and the auction of Washington wines, and then a few sources of Washington State University program funds. What happened to my advance? Oh, okay, now it works. pH is important because it's a major physiological factor that determines the evolution of the microbial populations during fermentation and during wine aging. To make this easier, and I think this is a mistake that I sometimes see made in discussions of the importance of high pH, we really need to consider these 3 stages of winemaking separate. There is the microbial population in the must and during the active alcoholic and malolactic fermentations. The next phase would be populations during aging. And then the final would be populations in bottled wine. Each of them have some constraints and some options that are unique to them, making it easier or a bit more difficult to manage things. I've said this a number of times, good winemaking practices is guiding the microbial population and closing spoilage windows, opportunities where a spoilage character might develop. pH, temperature, and SO2, starter cultures, and filtration are the main tools that we have as winemakers. Especially in these high pH musts that are more at risk of very rapid growth of all kinds of yeast and bacteria, including spoilage organisms, of course. It's very important that we exclude like the damaged fruit. And when you do a cold soak, make sure it's cold. Cold means under 10 degrees Celsius or around 40 Fahrenheit. If it's not cold, you're just incubating a whole bunch of microorganisms, many of them aerobic. You don't want them. You want to see that the alcoholic fermentation starts quickly, either right after you crush it or after a cold soak. So getting to the right temperature that encourages the desired population of microbes is really important. So temperature control and use of starter cultures, of course, is a great tool here to get the fermentations going. High pHs and some of the unwanted yeast populations can delay malolactic fermentation. Again, we can affect that by adding acids at the right time, as early as possible, of course. And of course, again, we can help with starter cultures here. Growth of microbes after the completion of fermentation is most of concern. I'll show you some data from our work there and some suggestions of what you can do. Managing the populations during, I'll first look mostly at managing populations during the fermentation and then the flavors that are derived from it. The goal of managing populations during wine aging is to keep them as low as possible. Really, after completion of alcoholic and completion of malolactic, we don't want any more contributions of microorganisms to the development of the flavor profile. The exception might be a wine where you want extra yeast lees aging, but that's a whole different technique that we can't get into right now. But in general, we keep microbial activity low by keeping the cellar temperature low, some racking, clarification as much as possible, and also SO2 additions, of course, are important there. The easy part comes in managing the pH and the populations is when you get the wine into bottle. I think all wines should be sterile filtered. Red wines at least cross-flow filtered to remove most of the microorganisms, yeast and bacteria. And the safest thing, of course, is to do this as soon as possible. As soon as the wine has aged sufficiently following fermentation, you want to get it into the bottle. And when you get it into the bottle, you want to exclude all remaining microorganisms. So that part is relatively easy and straightforward. A reminder, something that we've described for a long time, it's in all the textbooks. There's a magic pH that we have to pay attention to. It's around pH 3.5 in wines, below which we generally have Rhinococcus uni in the bacterial population, and above which we see more and more Lactobacillus and Pediococcus. Yeast, on the other hand, are essentially not affected by low pHs. They're happy even below 3.0, and there's no difference to them in terms of 3.5. We see higher yeast populations at high pHs around 4, but not a lot more in terms of diversity. This is a really important point to remember. This is looking at a lot of different wines. These examples came from Spanish wines, and it shows the importance of the real importance of pH 3.5 right here. In wines below 3.5, the concentrations of histamine, one of the major biogenic amine, is very, very low and of no concern. But as soon as the pH increases much above 3.5 or a little bit above 3.5, the concentration jumps up quite a bit. And we want to avoid those compounds in wine. They are unpleasant for the people and they really smell bad too, not histamine, but some of the other biogenic amines. There's a good side to managing that pH too. Here's a nice study that was done in Australia comparing malolactic fermentation in wines at low pH 3.3 and at high pH 3.7. And they could very nicely show how the pH impacts the fruitfulness of the fruit intensity in these wines after malolactic fermentation. A lot more fruit forward in the wines that started at low pH compared to the ones at high pH. And that experience is shown in a number of other publications. We generally get brighter fruit expression in wines that undergo fermentations at moderate and low pHs, not so much at high pH, which will be 3.8 and 4, and sometimes even above 4. So this is something to keep in mind. And of course, I have to remind you of SO2. One other reason why we want to keep the wine pH as low as possible always during must fermentations, especially during aging, and then in the bottle, is that we want to keep the total amounts of SO2 needed low as well. Just a reminder that the molecular SO2 that we rely on for its microbial activity is only a very small portion of the total SO2. at these high pHs here. And if we want to achieve even a moderate molecular SO2, 0.6 milligram per liter is a moderate amount to have an effective protection against unwanted microbial growth. At pH 4, we'd have to go to 100 milligrams per liter of free SO2. Nobody wants that in their wine, not a winemaker and not a consumer either. So we really, we do so much better managing SO2 effectiveness and with moderate concentrations if we can keep that pH low. Keep the pH low. The most straightforward thing, of course, is try to get an earlier harvest. We all know that the pH of the grapes increase during ripening, especially the later part of the ripening, as more and more malic acid gets metabolized in the grape berry. There are some other factors, potassium, of course, that might be accumulating under some conditions, fruit temperature. So, but in general, early harvest, of course, will give you a wine with lower pH and higher acidity. You might opt to harvest part of your wine a bit earlier than you usually do to preserve a bit more acidity. It also brings in a different flavor profile that might be quite attractive. So it's definitely something to consider. Tartaric acid, everybody is quite familiar with using tartaric acid as a means to lower the pH, and it's quite effective at that. It just brings an acid taste. Tartaric acid will also remove some of the potassium, which pushes up the pH. So you lose that as potassium bitartrate. But there's also an option of using DL malic acid. It's a food grade acid that's made synthetically, legal to add to wine. I was first introduced to that use actually in Australia when I was a cellar worker carrying bags of malic acid into fermentations. It's a good tool. You probably don't use it alone. I think a combination of tartaric and malic might be the best. The benefit of adding malic acid is that the D form of the malic acid is not metabolized. So that acid stays in the wine as acid, helping to control that pH. And the added L-malic acid is an energy source for the malolactic bacteria. So that keeps these bacteria happy during malolactic fermentation. Because these wines have such low natural content of L-malic acid. We also played around, and there's recently more interest apparently among researchers in the industry around the world using acid wine, a wine that's harvested very early with very high acidity, well over TAs of 8, pH is probably around 3, as a form of adding acid to the wine. The tricky part with that, of course, is that if we harvest these grapes so very early, they might bring in some flavors we don't want. So you have to keep that contribution, you have to manage that and keep the amount of acid wine or early harvest wine rather low so that it's acceptable with your final flavor balance. Another technology that works is ion exchange, but that's not available to many people. So the importance is try to aim for a pH that's close to 3.5 or below to start with. Then you can keep your initial SO2 additions low, and your final additions during aging will be more effective. We also looked at the use of starter cultures, yeast and bacteria starter cultures, and we looked at how does the timing of the malolactic starter culture addition impact the populations and the final flavor outcome. So you can do co-inoculation where you add the bacterial starter culture a day after the yeast starter culture. You can add the starter culture, the bacterial starter culture, of course, at the end of alcoholic fermentation. We call that sequential inoculation. And of course, temperature is really important. I mentioned that with the cold soak already. And during alcoholic fermentation, it's important to keep that temperature moderate, of course, also considering that along with the high pHs and low acid that we have, we also have high sugar content and high final alcohol content. So we definitely want to keep that temperature moderate and not let it go too high as the alcohol concentration increases, because that will increase the risk of stuck fermentations, the combination of high alcohol and high temperature. So we looked at tartaric acid and malic acid additions and the combination of the two. We had 2 years of experiments where we added an early harvest wine or an acid wine, and then the starter cultures. Some things I can summarize maybe right up front before we get into more data and more detail. We learned that pH is above to around 3.8. It's relatively easy to make these adjustments. It doesn't need large amounts. But boy, once you go over 3.8 at 4 and 4.3 that we've seen, it takes a lot of acid, either in the form of tartaric or malic and or acid wine. So there's some practical limitations. You want to not go much over that 3.8 harvest pH. Otherwise, you have to really be aggressive with acid additions to have a significant impact on that starting and fermentation pH. We also learned you've got to be careful with adding that acid wine, certainly above 10% as we did in one of the years. It had a pretty clear impact on the flavor quality of the wine, and it was not preferred. So it's better to keep that lower together with a lower starting pH. All these changes that we affected in must pH, either through acid additions or the additions of microbial starter cultures, did have an impact on the sensory quality. The first thing we can say is that starter culture additions work. The yeast additions and bacterial additions, we just follow the protocols that and the rates that were recommended by the manufacturer are very effective. We saw implantations of well over 90%, 95, 98% of the starter yeast and starter bacteria. So it worked. So this is certainly a very effective tool, even at very high pHs, 4 and even over 4. Without having to add acid, you can change the population right away. I'd encourage everybody to explore these tools, starter cultures, and the combination of these different acids for your own winemaking goals. In 2017, we did get some Merlot grapes from Sagemore Vineyard, and we did another experiment in 2018 with Merlot from Clipson Vineyard, each year shooting for pH in the must of around 4. We looked at the chemical composition of these wines during and after fermentation, and including sensory analysis. we looked at yeast and bacterial populations by traditional plating methods to see what actually was able to grow on selective media, and also looked at culture independent methods where we sequenced all the DNA that was in these fermentations, in the must and in the fermentations, to see what yeast and bacteria were present at any of these stages. In 2015, we did a first experiment, being pretty brutal in our additions of, or ambitious in our additions of malic and tartaric acid. We had a wine that started at a pH of almost 4.5, and we added tartaric, malic, and a combination of tartaric and malic pretty aggressively. We're able to get that pH down to that 3.4, 3.5. Although you can see pretty quickly that pH starts moving back up during the fermentation, and of course with malolactic fermentation. So the final pHs were coming similar again, although this 3.8 is still different from pH 4 and 4.1 in terms of the sensory perception. What else do I want to point out here? T8, of course, changes. That was the main thing. We added quite a bit of tartaric in this case, in that 3 grams and 4 grams of malic per liter. And just jumping to a flavor evaluation at the end in the wines that were about at that stage a year old. There were distinct, I call dirty aromas. They were like rotten vegetables, and there was some ripe, overripe fruit, not surprisingly, at a pH of 4.5 almost. The malolactic, the malate additions and the lowered pH gave us a pretty good fruit expression there and a pleasant texture. So the combination of malate and malic acid and tartaric acid. In this experiment, the tartaric acid addition didn't do as well. The very large addition we made was successful in pushing the pH down, but probably the remaining high tartaric acid content really accentuated the tannin impression. They were a bit too aggressive, too harsh. So that's something that, of course, you could change by making a slight deacidification, bringing that tartaric acid down a little bit. I was surprised how well the fruit was expressed in these malate and tartrate additions. For the next 2 years, we did things a bit more delicately, let's say, and looked at more diverse options. So in 2017, we brought our wine to that target pH of 4 by adding a little bit of potassium bicarbonate. We made additions of acid wine to bring to a target pH of 3.75 and 3.5. And we used 1 addition of tartaric acid to a target of 3.5 and 1 addition of malic acid to a target of 3.5 as well. But on top of that, we looked at no malolactic, we added yeast to all of them, and we added no malolactic to a third of the experiment. We did a co-inoculation and we did sequential inoculation in all these treatments. So the combination of starter culture addition and different acid additions is what we looked at in 2017. In 2018, we expanded this or changed it around a little bit. We still have an SO2, initial SO2 addition. But in this year, we also looked at completely no starter culture addition, no yeast and no bacteria. And that's what this is. No means no yeast and no bacteria. We had yeast and we had, oops, go back. Sorry, I hit the wrong button somehow, somewhere. We had no yeast and no bacteria added. We had yeast added and no bacteria, and we had yeast and bacteria added in all these treatments. To give you an idea of what amounts were needed in 2017, the amounts needed were pretty small because our fruit actually came in at a pH of 3.8. We didn't quite reach in 2017 the target of 4. So as I said, we actually added some potassium bicarbonate to bring the initial pH up to 4. It took only small amounts of acid wine, percent and 3%, to bring it to this initial target pH, and relatively small amounts of malic acid and tartaric acid too. In 2018, it was quite different. We had a fruit that was at pH 4.3, and we needed much more of the acid wine. Here in this case, and also more tartaric and malic acid. In all the experiments we saw, there was really no impact of acid additions and starter culture additions on the rate of alcoholic fermentation. When you at the high pH, you do get a larger yeast population. We saw that in each case here, but it had no effect really on the fermentation rate. They were all identical, as you can see here. And they were not affected by the pH and not affected by starter culture or no starter culture. This is just looking at the yeast population, total yeast population, and fermentation rates. When you look closely though, this is from the 2017 wine where we added starter culture. I highlighted the Hanseníaspera or Clucra yeast. They're quite present at the early stages of the must before fermentation. This is percent of total yeast presence, so between 20 and 75% of all yeast there was Hanseníaspera on that first day. On the second day, we still saw quite a bit, up to 90%. We added the yeast on the third day, and then checked again on day 6 here. The Clucra and Hanseníaspera yeast were very successfully suppressed, and we see Saccharomyces, and that's our starter culture there already between 82 and 92% of the total population. There's a whole bunch of other yeast you find there, but they're in percentages that are barely 1% of the total, except for like this Oreobasidium, that's a very common environmental fungus, has, as to our knowledge, no impact on wine quality, and it dies out pretty quickly during fermentations. Everything else is there at rather low populations as soon as the fermentation gets, as soon as the pH is lowered and the fermentation gets going. Big impact on the yeast, on the bacterial population. When you see a change in pH that doesn't seem very big, 3.8 to 4, we see quite an increase in the bacterial population. Now these are log numbers, log 10. So the change from 4 to 5 is 10 times more bacteria per milliliter than in the lower pH wine here. We saw also an increase here with the acid wine in bacterial populations. But then when we push the pH down, you can see very significant reduction of bacterial population in these musts. This is total bacteria. Here I selected out some of the major yeasts. As I said before, in that first must stage, there's quite a few yeast. These are the most common ones. There's this Oreobasidium again. There's Hanseníaspora that I just mentioned already. Penicillium, Torulaspora are there. But as soon as we add our starter culture and we have an active fermentation, we basically have Saccharomyces with a few of these Clechera or Hanseníaspora yeasts. When at the end of malolactic fermentation or after the end of malolactic fermentation, the yeast die off, you can see those green bars decreasing in size. And in reverse, we see these red ones increasing, and that's the Hanseníaspora yeast again, having a chance to grow back. And those would be the yeast that can produce some of these unwanted spoilage flavors, those rotten vegetable fermentations, higher concentrations of acetic acid and some bitterness. So this is the stage where we need to control this population, racking, passive filtration, and of course SO2 additions. If we are at pH 3.5, SO2 works well, but of course if we're higher, it doesn't work so well. I mentioned that already. The very slow effect that we saw on alcoholic fermentation rates with all these treatments. You do get a larger yeast population, but it has no impact on the rate of fermentation, but there's more biomass there for sure. Just some impressions on these wines from 2017. Again, we noticed there's some dirty, rotten vegetable fermentations, maybe sulfury odors in these high pH wines where we didn't try to lower the pH, and it's a little bit better with the bacterial starter culture added, maybe, but it still wasn't a very positive impression. As we go down in pH, we get much better fruit and better texture in these wines. So the acid wine, they worked quite well. The malic acid additions again worked really well. The tartaric acid addition was, maybe in some cases still a bit aggressive with the acid, but that was a young wine. Maybe with time that would have come into nice balance too. So acid additions definitely give you a different flavor profile. And this is looking at the actual pH changes that we had in 2018. So that was the really high pH must, about 4.3 that we started with. Really jumped up as the fermentation get going in the early stages of the more potassium presumably coming out of the fruit. It dropped during the fermentation, and especially where we added yeast starter culture. And then of course, it ended up at a pH about 4.1. So the impact of yeast only, there is a difference during the fermentation in the pHs. When we added the acid wine, we had moderate effect. We had some success in getting that pH down to 3.75. We didn't quite get the 3.5. It stayed down for a while, but by the end of alcoholic and malolactic, the pHs were pretty severe again. Tartaric acid, same thing. We were able to push down the pH initially. It stayed down, but as the fermentation kicked in and progressed, the pHs started rising again. Overall, the pHs in the tartaric acid addition stayed lower than in the acid wine additions. Was quite effective in keeping that pH under control. This is looking at malolactic fermentation rates. Interesting that the malolactic fermentation rate was impacted by the addition of yeast as well. So yeast actually suppressed some of those native bacteria where we didn't add bacterial starter culture. And that was true in the acid wine or tartaric acid addition, as well as in the control. Of course, where we didn't add the starter culture, it took much longer. You can see it took us over 20 days, actually around 25 to 30 days before the spontaneous malolactic fermentation was done. In this case, it took about 35 days for it to complete. Not surprisingly, when you're adding a starter culture, it gets going quickly and it's done quickly. We were done in 7-8 days compared to 30 days without starter culture addition. Here's that effect of pH on spontaneous fermentations, just looking at malolactic fermentation. So we added no yeast, we added no bacteria, just whatever was in that must. Control would be the high pH non-adjusted, and then a pH of 3.75 and 3.5. Of course, when we added the acid wine, we increased the concentration of malic acid with this addition. Looking at how fast malolactic fermentation ran, it actually got going and completed most quickly at the low pH in these spontaneous fermentations, followed by the next lowest pH, and then quite a bit slower in the high pH. This is all the native populations in there. So that the lower pH is really selected for those malolactic bacteria. And that goes back to that first slide I showed. At the low pH, you're strongly selecting for unibacteria, which are very well adapted to and love metabolizing malic acid in these low pH wines. They are not as competitive and not as fast growing at these high pHs. Of course, yeah, and I mentioned that already, the yeast addition also had an impact on the native malolactic bacteria population. It delayed it a bit, and it had brought out a somewhat different population. So here's a pretty color picture. I think it illustrates nicely. It gives you an idea of how diverse this population is. It's very easy to see that in these early stages in the must, we have a lot of different bacteria in there, all at pretty low numbers, except for maybe a couple or one group. And then that population changes dramatically as we get the fermentation going with SO2 additions and starter culture additions. You can also see that there is a different picture in a pH 4 compared to pH 3.75, the middle one, or pH 3.5. This is the total list of bacteria that we were able to find with sequencing in there. So very clearly, very different population dynamic just in response of pH. We can also trying to sort out what bacteria population went up, which ones went down. We always look for Henick-Coccus, of course. We would like that to increase quickly. And we saw that, by day 20, the spontaneous fermentation population got going. We have 25% and then quickly dominating to over 90%. Here in the acid wine, it was a bit earlier where we saw an increase in the native Henick-Coccus bacteria than in the high pH wine. And as a response, of course, we start suppressing lactobacillus and this fructobacillus, and also that lactococcus, all more adapt to higher pH and no alcohol. So it flips that picture. Henick-Coccus dominates, more alcohol is there. These other major players decrease dramatically and stay quite low. Here was a new thing to us, Catamella bacteria that have been only recently described in wines. Nobody has really figured out yet what they do. They're present significant numbers early on, and then quite a bit after in the later part of the fermentation, after alcoholic fermentation, malolactic fermentation. So that would be a question to look into more. What is the role of these bacteria that we find now with the new tools of sequencing and have overlooked, obviously, in the past with plating. Yeasts. This is the yeast addition only, but still looking at bacterial populations, looking at the control wine at the high pH, acid at acid wine at different pHs, and tartaric acid at 2 different pHs. Again, we see a large diverse population early on before the fermentation really gets going, especially in the high pHs. And then that shifts to more to the bacilli, and Henick-Coccus is in this group here, the red bars we prefer to see. And again, when you look at the detail, you see the similar picture with this yeast added and no ML bacteria added. Henick-Coccus then starts growing and outcompeting the other bacteria. Trying to summarize this in a simpler picture is, here we had no yeast added, no bacteria added, yeast added, no bacteria, and then yeast and bacteria added. Just looking at the major groups which were at lower population, red always means significant decreases as response to these inoculation practices, and the green and plus was a significant increase in the population. And I think this shows clearly that there are big differences in the general population, whether you add a starter culture and which starter culture you added. The biggest differences in the population was always in the early and middle part of the fermentation. At crush, they all started with the same population. And at the end of alcoholic, we had very similar microbial populations again, but there was, the diversity always happened in the early and mid part of alcoholic and malolactic. I just have a few more data on the sensory evaluation that we did and chemical analysis of the 2018 wines, where we had no acid addition to this must of 4.3. We had a tartaric acid addition, and we had the early harvest wine or acid wine addition. On top of that, we looked at the impact of malolactic starter culture, added or just yeast added to these two different acid treatments. Summarizing all the chemical analysis with the gas chromatograph and liquid chromatograph, the total number of compounds found to vary significantly was probably around 200, was well over 60. Taking this all together and then trying to find out whether this pattern of metabolite changes or differences makes any sense. Lindsay Konoski looked at that and did this cluster analysis. And it surprisingly actually shows that all these changes in the chemical composition that we were able to measure fall very clearly with the treatments we have. We have tartaric acid addition at pH 3.5, co-inoculated. We have tartaric acid 3.5, just yeast added, no bacteria. And you can follow over here, pH 3.5, early harvest wine, the co-inoculated and the yeast added again. They're close, but they're different. And then the control wines, no acid additions, yeast added or yeast and bacteria. Again, they're separated in these chemical profiles.
So, hey, Thomas,
Yeah.
we got about 10 minutes. So
Okay. I think this is probably number.
several questions. So
Okay, good.
you
This is just another look at all the data. It's a simple summary where I'd like to start with the sensory is when you look at these, the flavors, aromas identified by the taste panel, on top here are all fruit, floral aromas, spice, and tea. And then we go into pepper, yeasty, and vegetative, sulfury things. In this lower half, we saw a very consistent trend that with the in these less desired flavors here were predominant in the wines that had no acid addition and early harvest wine added. The wines treated with the tartaric acid addition always favored the expression of these fruity flavors. I think that's exactly where I want to end. Thank you. I'll take the slides.
Great. So I'm going to post some questions to you. And the first one is, can you touch on temperatures? Why not drop temperature to control bacteria?
Well, during malolactic fermentation, of course, you have to keep a temperature that's pleasant to the bacteria, which was between 70 and 75 or at the most 80 Fahrenheit. But yes, during storage of the wine, when you don't want any more microbial activity, especially bacteria are quite well inhibited by temperatures below 50 Fahrenheit. Does that answer the temperature question? I mean, it's not a perfect solution, but it does help. There's a number of storage of wine studies that have been done that show the lower the temperature, the lower the activity of microbes. On the other hand, your aging of the wine chemistry is also slowed down. So it's a trade-off.
Okay, and another one. Why do you think of these commercial non-sacs that are supposed to be added at the beginning of fermentation as a biocontrol, sometimes suggested to substitute for sulfur dioxide?
I think they work as a biocontrol, especially. I think it's a good tool in cold soaks. I'm not sure I would do it totally without sulfur dioxide. I would always favor still an addition of about 30 milligrams per liter of sulfur dioxide, probably together with one of those non-Saccharomyces as a tool to suppress yeast that are potentially producing bad flavors. Obviously, these selected non-Saccharomyces have been screened so that they don't make any unpleasant flavors. Yes, it's a good tool. And as this questioner probably also knows, if you want to complete that fermentation, you would increase the pH, the temperature then to around 65, 80, and add a Saccharomyces starter culture and get that alcoholic fermentation going and complete it without problems.
Okay, how turbidity of must will impact the ion exchange? Is it sufficient for must? At what stage do you recommend to use it?
Oh, that's out of my experience. We really need a chemist to answer that one. I would think a very turbid must is not going to work well in an ion exchange system. It's just going to clog things up. But what the cutoff points are, I don't know, but I could, Melissa will remind me to get an answer for that. Find somebody.
And then one question was, what yeast strains did you use? Did they degrade DL malic, or was it just impact of lactic of LAB?
We used EC1118, I think, in all these fermentations. It does not degrade any significant amounts of malic acid. So we always check the malate degradation, malolactic fermentation by following the L-lactate amounts as well. And we also looked at D-lactate to see if D-lactate was formed from degradation of sugars, but it was pretty much always made the bacterial conversion of L-malic to L-lactic acid.
Okay, and another question, do you know why it's not allowed to use sulfuric acid for pH adjustment?
Not for sure. I think it's just a safety concern. Maybe our regulators don't want people to handle concentrated sulfuric acid. But it's a great tool, but it obviously not for commercial winemaking. It's not allowed.
Okay, that is all the questions that came in. So Thomas, would you maybe ask for an interest, in the sensory sampling of wines? I know we have some that are from the Woodinville area that have joined us today. At the end of this, there's a survey, just a rating of how we did on the webinar. But I'm going to put in my email in the chat box. And if there were, if you can discuss a little bit of your idea on the sensory, if people are interested, they can email me.
Yeah, I'd be very happy to taste some of those wines we still have with wine industry members to hear your impressions and let you see what we were looking at.
So we were actually considering August 13th because Thomas will be over in Woodinville for a wine industry event. I have a wine commission board meeting in Woodinville, and we were thinking it might be a time he could bring some of these wines from this project over. And if there are some Woodinville or Seattle area winemakers that would be interested in sort of tasting through these wines, Thomas, we could set this up if there's interest. So
yep.
I have, so email me if you're interested, and I'll coordinate also with the Woodinville Wine Country. And Lynn Scott, I think he's still on, he's chair of the Wine Research Advisory Committee, and he can also help get the word out. So, one last question. Back with the histamines and pH, can we clarify which were red and white grapes?
In this summary, no, and it wouldn't matter. I mean, it's strictly looking at the pH of the wine and the concentration of histamine.
Okay.
Certainly, most of these wines would have been red.
Great. Let me make sure. I think we've got them all. Okay, with that, we're right at 4 o'clock, or pretty, yep, right at 4 o'clock. So thank you all And if you're interested in, Charlie Edwards is going to sort of give a culmination of his many years of work on Brett. He's now shifted focus and working on some new research concepts. So that is coming up in a couple of weeks in August. So with that, thank you much. And we look forward to sharing our research news with the industry at the next one. So
Thank you.
goodbye.